After the successful completion of the 9th chapter in the first PUC, let me start with the 10th chapter in first PUC, which is very important for your NEET examination. That is the cell cycle and cell division. So, cell division is the characteristic features of all the living organisms, and cell division is required for both growth and reproduction, etc. There are two major types of cell division: mitosis and meiosis. Mitosis is equation division because the daughter cells formed contain similar number of chromosomes to their parent cells, hence called equation division. Meiosis is such a type of division where the daughter cells formed contain only half number of chromosomes, that is called the reduction division. So anyhow, cell division in eukaryotic cell as well as eukaryotic cell requires as many as 24 hours and 90 minutes in the yeast cells, that is Saccharomyces. The cell cycle, it involves the interphase as well as the mitosis. It is interphase and M phase. M phase is called the mitosis. And 95% of this time, for example, if 24 hours means cell division, 95% of it will be spent during the interphase itself. So this in the interphase further divided into G1, S, G2 phases. So G1 phase is growth one phase. Precursors required for protein synthesis get mobilized during the G1 phase. S phase is very important. So S phase is called synthetic phase, where the DNA present in the chromosomes undergoes duplication. As a result, C2 becomes C4. That is, 2 chromatids becomes 4 chromatids. That is duplication of DNA results in the formation of such chromosomes which contain the C4 chromatids, that is C2 chromatids, C2 to C4, and then resulting in the formation of double-stranded chromosomes. And in animal cells, centrioles also divide. But the centriole duplication takes place in the cytoplasm, not in the nucleoplasm. So DNA duplication takes place in the interphase, that is in the nucleoplasm. As a result, double-stranded chromosomes appear, C2 to C4 appears. This is very important. Hence, the quantity of genetic material will be increased, and cytoplasmic ratio is maintained. That is the significance of mitosis. It is because of the duplication. Hence, the duplication plays an important role, and much time will be spent in this G1 phase, S phase, and in the G2 phase. The G2 phase is growth 2 phase, where the protein synthesis takes place. And the prophase. Prophase is the first phase in which chromosomes become thicker and shorter, and that is in the early prophase. In the late prophase, what happens? Chromosomes become still distinct, visible, and the nuclear membrane, nucleolus, disappear. Not only the nuclear membrane, nucleolus disappear, but also the Golgi complex, endoplasmic reticulum, nucleolus also disappears. This is very important, so far not given at the end of the prophase. During interphase, in some cells, for example heart cells, there is a G0 stage, that is the growth stage itself, G0 stage, where the cells do not proliferate. For example, heart cells, unless they are called to do so, they do not proliferate. This stage is called G0 stage, quiescent stage. And in case of plant cells, both haploid cells and diploid cells undergo mitosis. that is the alternation of generation is only haploidtic in algae and fungi. Majority of algae and fungi, thus their life cycle, that is alternation of generation, is said to be haploidtic type. That is, both haploid cells and diploid cells undergo mitosis in case of a few plants. That is in the plant cells. After the first prophase, what happens? With the disappearance of the nuclear membrane and the nucleus, chromosomes enter the equatorial plate called metaphase at the equatorial. Chromosomes, they are arranged in such a way that their arms are facing towards the opposite poles. Spindle fibers appear. This is about the metaphase in simple terms. And then the anaphase, very important. Very important. So here in anaphase, centromere divides. This is very important. Centromere divides, resulting in the formation of the sister chromatids. And these sister chromatids formed move towards the opposite poles. That takes place during the anaphase, which is very important. And in the telophase, chromosomes enter the poles. Nuclear membrane nucleolus reappear. In addition to again nuclear membrane nucleolus, Golgi complex and endoplasmic reticulum also appears during this. Actually, but the chromosomes lose their individuality, and the chromatin mass will appear. This is very important during the telophase. Thus, finally, what happens? That is about karyokinesis. Cell division involves karyokinesis and cytokinesis, and this is about the karyokinesis. And cytokinesis, that is the division of cytoplasm. Division of cytoplasm takes place in plant cells by cell plate formation. A bulged portion appears in the middle and extends towards the periphery, separating the two daughter cells. That is in plant cells. In animal cell, a cleavage appears, a furrow appears, and proceeds towards the center centripetally and separating the two daughter cells. And cytokinesis fails to occur. If cytokinesis fails to occur, this results in polyploidy. That is very important. Polyploidy, increase in number of the chromosomes, and then the separating the two daughter cells. In case of coconut, syncytium can be seen. Syncytium, free nuclear as the failure of cytokinesis. Syncytium in liquid endosperm of the coconut. So this is about the mitosis. The daughter cells formed contain same number of chromosomes. Qualitatively they resemble. Nucleocytoplasmic ratio is maintained. That is the significance of mitosis. Coming to the meiosis is called the reduction division. Daughter cells contain only half number of chromosomes. Here, only a single cycle of DNA duplication takes place, which is very important. And the interface, that is G1S G2 phases, the same as in mitosis, but the here the prophase is a prolonged phase. Further divided into first prophase is prolonged phase. First meiosis and second meiosis. First meiosis will have first prophase, first metaphase, first anaphase, first telophase, and the second meiosis will have second prophase, second metaphase, second anaphase, second telophase. Thus, there are two types of this one. First meiotic division together constitute a single meiosis. That is very important. So, first prophase is a prolonged phase in the meiosis. First meiotic division. Further divided into LZPDD, leptotin, zygotin, phacotin, diplotin, diaconasis. LZPDD code word. Leptotin. During which the chromosomes are placed, double-stranded chromosomes, so are placed towards one end, and it is called bouquet stage, appears like bunch of flowers. And during the zygote, pairing between two homologous chromosomes takes place, that is called synapsis, pairing between two homologous chromosomes. This results in the bivalents, which is very, very important. And they are called tetrads also, but they are invisible at this stage of the zygote. It is the pairing between two homologous chromosomes. And at the same time, this pairing together forms a synaptinemal complex because of the appearance of a protein called synaptinemal protein. Synaptinemal complex will be developed during the synapsis. And next stage is the Facetin stage. And one important thing is leptotin and zygote short-lived. This is important. And then after this, LZP, Pacetin stage. And the tetrads become more clear appearance of these tetrads. That is also very important. And the recombination, that is crossing over, occurs during the Pacetin stage, and exchange of genetic material takes place. That is because of an enzyme called recombinase. Genetic recombination takes place. Crossing over takes place during the Pacetin stage. And the first stage of meiosis, first stage of meiotic division is prophase 1 is the Diaconasis. The first stage of is diplotin stage. So the chromosomes which have undergone crossing over tend to move apart. As a result, X configuration appears, called chiasma formation. Hence, this is the first stage of the meiosis. The final stage is the Diaconasis, wherein chromosomes become thicker and shorter, and nucleolus, nuclear membrane, along with endoplasmic reticulum, all these things, Golgi complex disappear. And terminalization of the chromosomes takes place in the diplotin, in this stage, in this Diaconasis, terminalization takes place. And in diplotin stage, one point I have forgotten, that is in case of oocytes of some organisms, the oocytes of some diplotin stage lasts for many months. That is in diplotin. The final stage is Diaconasis. Next to this, the first prophase. After first metaphase, chromosomes surrender the equatorial plate with the disappearance of the nuclear membrane and the nucleolus. These chromosomes are arranged in such a way that their arms are facing towards the center. That is very, very important. And spindle fibers appear naturally. This is about the first metaphase. Coming to the first anaphase, here this stage is very important. Centromere doesn't divide. This is very, 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 very important. As a result, half number of chromosomes, not sister chromatids, half number of chromosomes move towards one end, half number of chromosomes move towards opposite end, and nucleolus and nuclear membrane reappear during the telophase stage, and cytokinesis takes place by cell plate formation or the furrow formation in animal cell, and two daughter cells are formed called dyad cells. These dyad cells, by skipping the interphase, hence I told you earlier, only single cycle of DNA replication takes place. By skipping this the interphase, enter into the second prophase or second metaphase sometimes. During the second prophase itself, again the chromosomes become thicker and shorter, all those things, and it is nothing but the mitosis, second meiotic division. Hence, in the second metaphase, chromosomes arranged in they are the equatorial plate arranged in equator. They are arranged in such a way that their arms are facing towards opposite poles, spindle fibers appear, and in the second anaphase, centromere divides as in the mitosis, and then sister chromatids here move towards opposite poles, and then nuclear membrane nucleolus reappear, and the cytogenesis takes place, separating four daughter cells. The stage between the first meiotic division, second meiotic division is called interkinase stage, that is in between the first meiotic and second meiotic division. Thus, four daughter cells are formed. Thus, the increase in the variations and decrease in the number of chromosomes paradoxically can be seen. This is the significance of meiosis. This is about the cell cycle and cell division. The major differences between mitosis and meiosis: equation division, reduction division takes place both in reproductive cells as well as the somatic cells, and it takes place in the germ cells only, and sometimes in lower plants it takes place, and then the required for both growth, reproduction, and required for growth, etc. And first prophase, there are no two such subdivisions like here in mitosis, and two subdivisions: first meiotic division and second meiotic division. No bouquet stage, no crossing over, no synapsis. Synapsis takes place, no chiasma formation takes place, and then only two daughter cells are formed, four daughter cells are formed. These are the centromere divides in the metaphase, and centromere doesn't divide at the first metaphase, but it divides at the second metaphase, that is in second meiotic division. So these are the major differences between mitosis and meiosis, which is very important. And the significance of mitosis is nucleocytoplasmic ratio is maintained, and here nucleocytoplasmic ratio not maintained, increase in the variations and decrease in the chromosome number. That is the significance of the meiosis. So with this I have completed the 10 chapters that is in first PU, and all the chapters in second PU, giving importance to the NEET type of questions. Please subscribe.